For those of you here in person, thanks for joining us online. For those of you online, you're missing some good pastries and coffee and swag, but maybe you'll learn your lesson, come in person next time. But no, I know people can't all get here this morning. So I'm going to give people a second to settle down. Okay. Well, good morning and welcome. This is the grand opening of the Biomedical Informatics and Data Science Department. We have had a number of false starts due to various factors outside control of the university, but we finally got approval, the final approval from the state of Alabama on January 1st. And so we're been scurrying around trying to get all our ducks in order. And so the former Informatics Institute is now Department of Biomedical Informatics and Data Science. So this morning, I'm going to give some introduction and talk about what we're up to, history, current activities, and vision. Dean Agarwal is going to give some welcoming remarks. We'll hear from Jake Chen about his new Spark Center, and we'll also hear from Jin Chen about our, I don't know what that word welcome is doing there. We'll hear about our educational programs that we're starting up. Okay, so a little history. So 2014, Dean Selwyn Vickers got approval for establishing the Informatics Institute. I came in 2015 and started recruiting faculty, staff, and trainees, and there's been various growth spurts over the years, but that's our current status. And then, as I said, Dr. Agarwal now has established our Department of Biomedical Informatics and Data Science. So, current activities. Well, we're like every department. We do research, we do service, we do training. And so for our research, we work a great deal with the Center for Clinical Translational Science. In fact, it's often difficult to tell sometimes which unit is doing what. There's a lot of overlap. So we're involved in overseeing the bioinformatics analytics. And let me just get through this list here. We have the Alabama Genome Health Initiative, all of us, electronic medical records, eMERGE, and these are, let's see, can I go backwards here? It doesn't seem like I can. So we'll just stop. Oh, maybe I can. So these are all large grants that the Informatics Institute has played a key role in obtaining funding and carrying out the mission. And then the faculty in the Informatics Institute have been doing their own research as well. So I looked back at our CVs over the past 9 years, and we've had 90 grants, 245 papers, 120 abstracts. So we've been pretty busy. I'm not going to try to cover all of that research in this talk this morning, but I'll show some highlights. Our education, our current education, we have a Bachelor's of Science in Bioinformatics that we established with the College of Arts and Sciences. And as of this year, we've had 8 graduates. We have currently 55 students enrolled, 25 are continuing, 30 new. And you can see the demographics of our population. So that's a thriving program. It's been growing each year, led by Elliot Lefkowitz. And then we have a Masters of Science program. We have a Clinical Informatics Master's track in the Health Informatics program in the School of Health Professions. We have a Bioinformatics PhD in the Genomics, Genetics, and Bioinformatics theme in Graduate Biomedical Science. And we also have a Bioinformatics PhD track in Biomedical Engineering in the School of Engineering. So that first PhD is more biology-oriented, and the second one is more computationally oriented. Current service, as I mentioned, bioinformatics, analytics, a lot of that is done with folks in the CCTS, but some of our faculty also collaborate with other researchers to get their data analyzed, particularly Jake Chen, Sei-Chen Chang. And just that's the CCTS publication record, but we have some overlap in that area. We also run the I2B2, and we help provide access to data in the Enterprise Data Warehouse. So the Enterprise Data Warehouse is basically a copy of the Cerner database, and it's got, as you can see, the statistics, 1.8 million visits. We've got 707 million observations and 345,000 unique patients. And then we have, there's the patient volume, we have 200, sorry, that was just last year. So total, I thought it was a little low. Yeah, we've got almost 10 billion observations now in the database. I2B2, if you don't know what I2B2 is, I've been talking about it for 9 years, so if you don't know what it is by now, you won't know after this. But it's a user-facing front end that provides access to a great deal of the data that's in the enterprise data warehouse. Not all of it, but it's a self-service system that lets you go through huge terminology to pick out terms of interest, create a Boolean search strategy, and then select a set of patients. So in this case, I've selected, done a little simple query of ending two concepts together, and I got 30,000 patients. I have a choice. I can download a limited data set on those patients, or I can go to the warehouse analysts and they can use that cohort then to get a fuller data set with IRB approval. But the download of the limited data set is automatic, which is pretty unique for I2B2 users around the world. The terminology inside I2B2 is powered by something called the UAB Foundational Ontology, or UFO, and that's as a starter set, it's all the terminologies that are used to code all the data. And I don't expect you to read this, just expect you to see that there's a lot of stuff in there, and this is just the disease section. And if we said find all the patients with respiratory infections, if you were to go into ICD-10 and SNOMED and the other terminologies we have, things are scattered all over the terminology, and so you can find them if you're persistent. But the idea of the UFO is to create a single ontology that unifies all these things and makes it easier to find things. So, for instance, in this case, we create a class of respiratory infection, and then that subsumes all these other terms that we might be interested in. And Amy Wong is our resident ontologist, and she's managing that. The UFO, Foundational Ontology, is going to be key to a lot of other things that we're going to be doing. We provide clinical data access to UAB users, that's through I2B2, and here are some of the statistics of user requests and the data sets that we've been getting, and we'll make these slides available. You don't have to try to read all these numbers. And then we do sharing outside, so we share with a number of different national programs. N3C is the most recent, and that's the National COVID Cohort Collaborative, and that's the NIH's database to bring together data for all patients tested for COVID-19 from partner institutions. UAB was one of the first 4 institutions to contribute data back in 2020, the beginning of the pandemic, and we continue to contribute data there. And we contribute data to the All of Us program, and we manage the Cooper Green database as well. We contribute data to One Florida and Trinetics and Spark. So these are all collaboratives that provide access to our data. Either we send them data or we provide access for on live queries. We do a lot of data sharing, and that's originally through the Shrine Network, but that has been subsumed by the ACT Network and now the ENACT Network. These are all NIH-funded collaboratives to provide access basically to our I2B2. So someone, you all, can log into the ENACT Network and do a query that looks like an I2B2 query, but it queries all the different members of the ENACT Network and brings together summary data from all these sites. And then what you can do is say, oh, this site has a lot of patients that I'm interested in. I'm going to go find a collaborator at that site and see if we can establish a program to enroll their patients or use their data retrospectively, what have you. Trinetics is a commercial enterprise that does similar things, bringing together I2B2 and other data repositories. And then we're part of the One Florida PCORI network. And most recently, Northwestern has spearheaded a collaboration called Critical, which is creating a database of ICU data of lab, vital signs, medications, and de-identified notes, so people can go and query those data for retrospective research. And I mentioned N3C. So the National COVID Cohort Collaborative, you all as UAB folks can log on to this. You can get access, and you can establish your own research program. You can get collaborators from UAB or from other sites that have joined the collaborative. And in here, you don't download the data. It's an enclave. So you go in, you identify the patients you want using their ontology, you create a data set, and then the analysis occurs on their site, and you can download the results. We have at UAB, the last time I looked, we had 15 enclave projects, 74 members, 27 local investigators, and 45 project memberships. And we have a couple of awards that people have gotten in order to do this work. We've had 40 publications, 31 local authors, and there's our network. You can see UAB there, and centers around the country, and also some in Canada that we're collaborating with. Okay, we do natural language processing, and I think John Osborne is leading that work, and it's particularly exciting now because of the emergence of generative AI and large language models. So that's opening up a whole new area for research. And one of the things that that John has been doing is this virtual patient registry. It's called FEDERS, a Phenotype Detection Registry System. So if you are interested in a particular condition, you can create a virtual registry in FEDERS that will monitor our EHR and other registries to find patients that might qualify for your study. And it uses ICD codes and sort of typical things, but it also does natural language processing on reports. And one of the things I just learned that was very interesting to me is that John's system is looking for evidence of condition in reports using natural language processing. And some of that evidence is what we call incidentalomas. Those are findings that a radiologist notes on an x-ray that they weren't looking for, but they say, oh, by the way, the patient also has this other problem. So somebody comes in and say a gunshot wound, they go, oh, the bullet's here, and these structures are damaged. And then they say, by the way, the patient seems to have a tumor in their lung. Well, the people in the emergency room treating the gunshot wound may not be paying much attention to the tumor in the lung, and sometimes those things fall through the cracks. And so this project is not just helping to create a registry to say, hey, here's somebody who might have cancer in the lung and you might be interested in. They can also do follow-up with the patients and their physicians to say, hey, make sure you follow up this thing. And they're actually helping people to recognize that they have conditions that would otherwise have gone missed. That's pretty exciting. So the big picture, lots of pieces. We have a lot of things providing data, going into our repository, into the Enterprise Data Warehouse, which is really a Cerner database, and also I2B2 has other data, for instance, social determinants of health and disease, data from other research projects like the Alabama Genome Health Initiative and so on. And then we provide access to other networks and for our users to get access to their data and their researchers to get access to our data. Okay, so the vision. This was the Institute vision, and it seems like it still works for the department. We envision sharing of data between biomedical research and patient care that results in greater knowledge and improved health for people everywhere. So sharing of data seems like a pretty specific thing, but that's really the key to everything else. Because once we have data, then we can start doing analysis, we can generate knowledge, and we can figure out what those data mean and use them for improving health of people everywhere. Our mission, if it's going to come up, there we go. From data to knowledge to better health, our researchers will help apply their tools and techniques to problems facing researchers, practitioners, and patients at UAB and its partners, conducting our own hypothesis-driven research to develop tools and train informatics researchers of tomorrow. So that's a key that a lot of people don't realize, that informatics is not just applying a bunch of tools that somebody developed to a problem that they're trying to solve. It's also developing the tools, and it's also developing the theories and the hypotheses that help us develop those tools. So think about if you know what BLAST is. BLAST is just sort of a puzzle solver. David Lippman at NCBI developed it with his team. And it's really just a puzzle solver, but you can use it to solve some important biomedical questions like gene sequences, protein sequences. And so that's the kind of thing we do in the Informatics Institute, now the department. So our plans, recruitment is really the key. That's the thing driving me right now. The first thing I have to do is recruit all my institute faculty into the department, which is in process. That should not be too hard. So there we are as of today, and I've been charged to recruit 8 to 10 faculty over the next 5 years, which I take to mean recruit 10 faculty. So they want to give me 10 slots, I'm going to use them. And this is just sort of a rough projection, but if we have 18 faculty, maybe we'll double our staff, we'll triple our trainees, and it's going to be a sizable group. You're going to hear about this Jake Chen Spark Center, and some of the people that he recruits will also be in the department. So faculty, staff, trainees. So we really don't know how big we're going to get, but we know we're going to get big. once we've done the recruitment, then we can start doing all the other things. You know, we have a small faculty right now. So when you want to do natural language processing at UAB, you have to go to John Osborne. We need more people to help him. When you want to do ontology work, you have to go to Amy Chen. She's the only one or Wang, I'm sorry, who is working on the ontology, but it's a huge task. So and then we're going to start with our degree programs that you'll hear from Jin Chen later this morning, and we're going to need more faculty to teach those programs. So education plans, just to say, we're in the process of developing a PhD in biomedical health informatics. We're working with the School of Health Professions and Health Informatics, their health informatics program, and with the Center for Biomedical Innovation, doing an artificial intelligence in medicine track within our PhD. And we'll also be developing postdoctoral training programs. All right. For clinical and translational research, we're focusing a lot of our work on something called UBright. And UBright is our platform for team-based translational science. And the first part that we're fleshing out now is Data Lens. Sorry, that's UBright, not IT, but Data Lens is the platform where you now will go, if you want clinical data, we're phasing out the access to I2B2, and you'll be going into Data Lens to establish your data sets, but you'll also have directories where you can look for tools, for people, for external resources, computing resources, storage resources. All of those will be accessible from within UBright and Data Lens so that you can come in, establish a program, get your data set, and work with collaborators in that environment. So, as I said, we'll share the slides. This is the current view of UBright and all the functions that it provides. And again, if you're a UAB researcher, you can get access to this and check it out yourself. All right, clinical informatics. There's a big push now, initiative in UAB medicine for the learning health system, and the informatics department is going to be a contributor to that. So if you haven't heard of the learning health system, it's basically, the idea is a cycle of care, research, and innovation to improve patient health. So we start with patient care, data getting entered into the electronic health record. We pull those data out, give them to researchers, we generate knowledge, and then we take that knowledge and inject it back into the workflow. And it seems like, well, sure, that's what, we've been doing this for decades, centuries even, with paper records. So, what's the idea? Well, the idea is that now, with the ubiquitous presence of electronic health records and all the data that they contain, it's really increasing the opportunities with big data that we have in patient record and also genomic data, public health data, and so on. And so, we can build now applications that say, hey, we need more data in the record. Let's put, for instance, Fitbit data or data that patients collect at home on their glucometers or what have you. And then we have to get those data out, give it to researchers, and then when we want to put knowledge back into the workflow, we have to have a way to do that. So we work with health IT folks to do those things, but right now, each of those is sort of a one-off project. So, we're going to add these data. We're going to add the genome-informed risk assessments from the eMERGE project. Well, that's a 6-month project to build an interface to get the data into the record. And, there are standard interfaces now, FHIR and so on, but there's still a lot of work that has to be done to define what those data are, where they go, and then how you get them out. You need another program to do that. And then if you want to change the workflow, you've got to, you need another program. We want to use informatics to bring together and create single points of contact for data entry, data extraction, and knowledge injection. And we'll use the UAB Foundational Ontology as a knowledge resource to do that. And the idea then is somebody comes along and says, I want to put more data into the record. Well, you define it in the UFO, and that interface then will know about it and know where to put it because of the way it's defined. It doesn't require 6 months of programmer time to develop a new interface. Similarly, to get the data out, if the terminology that's used to code the data is in the UFO, you can use the Data Lens to extract the data. Injecting the knowledge into the EHR, into the workflow, that's going to be a tricky one, and one I'm looking forward to. For instance, we're working on a project now. I'm working with John Osborne, Tiago Colicchio, and others to develop a semantic model of the patient's record, all the facts in the record, and then create a navigational tool that will allow you to look at, for instance, a problem. And this is, again, hard to read, but each of the horizontal bars is a different problem in the patient record. The vertical color are different events in this patient record. So the pink is an inpatient event. And then you can see medications and labs and other procedures that occur, and zoom in and navigate around through the patient's record based on the semantics of what's in the record rather than just the chronological events. Okay, now we're going to hear from Jake Chen about the Systems Pharmacology Artificial Intelligence Research Center, which is another new initiative of a School of Medicine. And I'm not going to talk about it. We'll hear from Jake later this morning. And so our collaborations, I've gotten just a few of them on here within the Department of Medicine or the School of Medicine. Some of the departments that we've worked very closely with, some of our faculty are in those departments, and they're now moving into the Biomedical Informatics Data Science Department. And then we've had collaborations and continue to have collaborations with multiple schools. And I probably missed some in this diagram. But when I was recruited back in 2015, I was told UAB is a really collaborative place, and it is amazingly collaborative. It's been really easy to work with folks in all these different units. So, in conclusion, UAB, it's time for UAB to join the big guys and get a Department of Informatics. The plan is to be one of the leading departments in the field in the country, and certainly the biggest one south of the Mason-Dixon line. So the Informatics Institute has laid the groundwork. We've gotten grants, we've developed collaborations, we've got faculty, we've got training. So it's all fertile ground for building. This reinvestment will allow us to expand the faculty, and then the expansion of the faculty is going to lead to better research, training, and service. And then the collaborations that we have reflect the interdisciplinary nature of biomedical informatics. So, there isn't really a good, we talk about biomedical informatics, and then you got to say, well, what about, public health, and what about health service research, and all these other fields. And so by creating the term biomedical informatics and data science, we hope we're inclusive enough to make people understand that we're either working in these areas or collaborating with people that do. So stay tuned. It's an exciting time. I mean, even in the last year, we're seeing huge changes with these things like these large language models like ChatGPT and so on, are really going to change all aspects of what we do. And so it's an exciting time to be investing in informatics. So watch for what we're doing in healthcare, clinical translational research, and education. Okay, I have a lot of thank yous, and I'm going to forget people. There's folks that have really helped us create the Institute, people that helped me with the transition from NIH to UAB, and I'm leaving people out, I'm sure. But I also want to be sure to thank my faculty and staff, have been just a tremendous group to work with. Jake Chen, the Associate Director of the Institute, and all the faculty that we've had over the years. A couple have moved on to better things. And Jeff Gordon, Matt Wyatt, who do our technology arm, and then Brian McCain, communications, Jin Chen, you're going to hear from, and Ryan Melvin, who help out with education. And especially Amanda Carballo, who's you've met at the entrance there, who put this all together, done a tremendous job. So I want to be sure to thank everybody that has really helped us with this. So I'm going to lead a round of applause for that. And that's it. I'm going to now turn it over to Anupam, but let me see, I probably have time for a few questions. I've lost track of the time, but I think I was using my New York voice, so we probably have a little time. Any questions about the department, the institute, the transition, job opportunities? Hopefully most of you already have jobs at UAB, so if you know people that don't have jobs at UAB that are looking, let me know or let them know. We're going to be hiring faculty, and then from there, staff and trainees opportunities will follow. Okay, well, thank you. And with that, I'd like to introduce Anupam Agarwal.
Thank you, Jim. Really appreciate the great introduction to a new department. Good morning. I'm truly honored here to celebrate a major advancement for the Heersink School of Medicine, and I think for UAB as well. You know, you heard a little bit from Jim, my predecessor, Dr. Vickers, Selwyn Vickers. When he became dean in 2013, in the fall of 2013, he had three main priority areas that he wanted to, be part of our strategic plan. And those three were, at that time, precision medicine, genomic medicine, and biomedical informatics. So within a year of his being here in June 2014, the Board of Trustees approved, three institutes, as you know, the Hugh Call Precision Medicine Institute, the UAB Hudson Alpha Genomic Medicine Center, and, most importantly, the Informatics Institute. And within 6 months, through efforts of Dr. Kimberly and others, we were able to recruit Dr. Jim Cimino. He was the chief of lab for informatics development at the National Institutes of Health Clinical Center. So he was recruited here as the inaugural director in 2014. To recruit someone like Dr. Cimino really was a huge endorsement of our potential to be a leader in the field of biomedical informatics. I was executive vice dean at that time with Selwyn Vickers and was closely involved in the development of the institute and really privileged to work closely with Jim and his team in growing that institute further. Really, the next step in its evolution, following an external review by really three key leaders in the nation, the key recommendation was this needs to be a department to make it, any substantial gains. So we worked with Jim and his team very closely, and as you know, January 1st, we launched the new Department of Biomedical Informatics and Data Sciences. So really the key goal was to allow for growth of faculty to really be part of our tripartite mission, to be able to, contribute to service, to research, as well as education. You can do that a lot better in the context of a department rather than an institute, and that was really the rationale for developing it. And you can see very ambitious plans in terms of recruiting, faculty, recruiting students, and so on, that you will see in the coming years. I think in research, this is going to play a key role, in the next few years. Analytic tools with big data, biomedical informatics, translating to the learning health system, as you just saw with Jim, that's going to be very, very important. And Jim has, presented to our health system board, to our practice plan board, uniformly, there's been great enthusiasm that this is a huge unmet need that this department would fill. So we are truly excited in that regard as well. I think really like to applaud Jim and his team for having worked tirelessly and patiently through this transition. We've had a few bumps in the road with some policies and procedures that we had to complete to name this department formally as of January 1. I think, again, you heard about collaboration, and more than that, I think it's the spirit of innovation as well that this department could really, going forward by that bold vision statement, looking forward. I'm truly honored to have been part of this journey of transitioning this institute to a department and cannot wait for the next few years of the excitement that is forthcoming. So Jim, again, congratulations. Congratulations to you all. It takes, a big team to put things together. And we're looking forward to you all moving to your new home as well, hopefully later this year, sometime around December or early next year. So a lot of excitement. Thank you all very much for being here and looking forward to hearing on the next presentations. Thank you, Jim.
Thank you, Anupam. So when I was recruited here back in 2015, the first person I met was Bob Kimberly, and that was a social part of the visit, and it was at Bottega, and he introduced me to the craft brews of Birmingham. And I was so relieved to find out that if I came to UAB, I wouldn't be drinking Pabst Blue Ribbon for the rest of my life. But the first person that I met in the actual interview process was Anupam. And I'll never forget sitting in his office and looking out the window over the city. And I've never been to Birmingham before. And hearing about all the things that were going on. You know, I've heard about UAB. I'd done my homework, you know, before I came down when the recruiters contacted me. I'm like, UAB, I've heard, you know, I knew Ida Berner, the late Ida Berner. I've known her for decades. But I really didn't know too much about the place. And I remember Anupam filling me in and telling me really impressive things that were going on here. Got me very excited about the visit that followed. Okay, so I think next we have, is it Jake? Jake, are you up next? Yeah, so Jake Chen, Associate Director for the Institute, and now we haven't figured out what he's not the Associate Director of the department. We got to work out where he's at, because now he's got his own center. And the Spark Center will be inside the department, and very excited to hear about what he's going to be doing. So Jake, take it over.
All right. Well, good morning. Well, really appreciate Jim and our Dean Agarwal for introducing the department. And what I'm gonna actually talk in the next half an hour is launching this center called Spark. It's been planned after maybe several months and 6 months, I think. Throughout this process, and many people helped, and especially CCTS, Bob Kimberly, and many members in my lab, AI.map, and many enthusiastic collaborators in the School of Medicine. So I'm very fortunate to talk about it. So we do have a website now. It's called smartdrugdiscovery.org. You can go there and read the very basic information about Spark. Spark is a research center. We're not trying to become a pharmaceutical company yet. We want to combine several amazing technologies. One of them is, of course, artificial intelligence. And then two other technology that we believe are very critical to this center is systems biology and quantitative pharmacology. So we want to do research through innovation, and we also want to engage many potential partners in the School of Medicine, both from the clinical side and the basic science side, and then also reach the collaboration through academia and industry partnership. So what is Spark's mission? Is peace. What is peace? Peace is putting together the three words that's key to our mission to revolutionize the future of precision drug development. One of them is we want to make it personalized. And also we want to accelerate the process of drug discovery and make it economically accessible for all. So what do I mean by this? If you look at the statistics, someone did the review of average cost of drug discovery. From 20 years ago, it was about $800 million, $900 million dollars on average, if you factor all the failed drugs related to introducing one approved, FDA-approved drug. Now, 20 years later, remember, this is in the backdrop of the genomic and nanotechnology and many, many technology that's amazing. And then the cost actually just increased threefold. And when you look at the cost breakdown, about 40% of the cost in drug discovery is spent in the preclinical, right, in the discovery. and the 60% is spent on clinical trials involving patients. So you can say that a certain part of it, clinical trials, being so expensive and involving doctors and patients, and it's almost a fixed cost. And it seems to be a mission impossible. In fact, many research communities are coming up with new ways and thinking about this accelerated discovery. For example, American Society of Clinical Oncology, the one that I'm actually serving on the grand review panels, come up with this grand scheme. On the left side, you take measure of the traditional patient records and information, everything about the tumor, everything about the history, clinical records, but make it more multi-modality, right? So collect as much information about imaging, pathology information, genomics, and all of these information are fed into a so-called mathematical model. It's like a magic black box, and somehow on the other side, we can come up with drugs. And this model really works in getting some kind of a drug or a drug into the market. For example, with this model, we're seeing more and more cancer coming up with novel drugs, and not only the small molecule drug, but also immunotherapies and maybe even radio-guided chemotherapy drugs. That's a new kind of a modality coming out. But is this the solution to the problem that I just raised earlier? Right? So I think it's not that easy. It's not that easy is because, first of all, the data are not just a single modality. You have to think about drugs and the genes and diseases and their relationship all together in this context. And also because we're working in this translational space. In this space, some people are working on cell lines, and because they think that cell lines are easier to manipulate. Some people are actually putting things together into animal models because at least you can do experiment on them, except that they're not humans. And some people are actually working on the human model. They're very accurate, however, we cannot actually use human for all the experimentations. So in this space, and it's turned out to be very expensive and very slow. And also, in order to achieve the knowledge, I believe that we have to actually work together. Just having the biology, the basic biochemistry and cell biology is only part of the quadrant. And maybe there has to be a chemistry knowledge, and how does molecular shape translate into dockable molecules that can inhibit or activate proteins. And also you have to understand the medicine, right? What kind of a patient, having different complications, and how do you enroll patients into trials, and what are all the different kinds of conditions that the patient may have in addition to the disease of interest. So in this regard, I think that the disrupted drug discovery really requires on the data side, the data collected has to be multi-scale, multi-modality. And in the model side, and we have to come up with not only these existing model, but also digitize them into in silico model that we can do many, many perturbations or tens of thousands of times in a second. And also on the knowledge side, I think we have to work together and then somehow share our knowledge in a entity. And this is actually why I think the Spark is so important. Spark is located in the Biomedical Informatics department, but I hope that it will thrive in the fertile ground of the entire UAB, because we would need all of these disciplines. Now, even with that, then what is the current dilemma and what is the future promise, right? So the current dilemma, I wouldn't read this entire table, you can read, that the trial target usually is a patient cohort. The time frame at drug discovery can take 8 years. If you break it down, it's every step takes 1 or 2 years. And the stages is very sequential. You have to first come up with in silico models, then go in vitro, ex vivo, preclinical, clinical, phase 1 to 3. You cannot skip step because FDA doesn't want you to skip step for reasons of safety concerns. And the cost is very expensive. Output, on average, we're only getting about 50 new molecular entity approved per year, and that has been stagnant over the past 10, 20 years. And the clinical trial success is hovering around 10%, and the side effects usually is unpredictable, uncertain until you have it trialed. But would there be a future possibility? I'm not saying that we're achieving anytime soon, but this is probably a problem to think about for all of us. Can we actually do drug discovery on individuals? And one, short time frame, days, integrated, directly go from in silico model to trial. You may say it's crazy, but maybe we haven't thought about that. The cost could be several $1,000s. And the output, how about more than 5 million possible combinations of therapy per year? Essentially, every patient would have its own specialized, accelerated N1 trial and development, where the success rate is 90% and the side effect is completely predictable. Now, if you want to think about how crazy we are, we have to actually do the disruption X-fold in each of the categories. And even the smallest category require 10-fold improvement, productivity improvement and increase. So obviously, this is not something that we can do incrementally. We have to actually do something really outside of the box. So a vision for Spark is the following. We want to revolutionize searches of new disease drug targets, drug modalities, and treatment combination strategies through systems pharmacology, AI, patient digital twins, and multidisciplinary partnership. Right, so I want to give you some sense, what are the drug targets? The targets are actually the proteins where the molecules are directly trying to unlock. They're drug targets. You have to understand the biology in order to design them. And then the modality usually is the engineering approach, try to perturb the target. And sometimes you have to use a small molecule, sometimes you use an antibody. Sometimes you have to use what is really popular last year and so far in January is called antibody drug conjugate, where you put the equivalent of a GPS antibody with a linker together with a chemotherapy drug, so you can specifically find the tissue and then just kill the cells for cancer. And these are all different modalities. And then the strategy is actually what I think is very few people are talking about. The strategy with the popularized by AI, we can think about just fighting a disease as a multi-pronged process, not just throwing one atomic bomb and then all of a sudden all of the cancer cells are killed, and then there may be a lot of collateral damage. The strategy may be sending some surveillance troop and to try to figure out the state of the disease, and then you send in grenades and tanks and then laser-guided missiles, one after another in waves, and some of them are actually existing tools in order to fight against a cancer in a war situation. So, for example, let me just show you one example. So KRAS is a successful drug target in treating many types of cancer, including pancreatic cancer treatments. And KRAS is an oncogene that actually normally would receive the extracellular signals and try to convert it into MAPK kinase signaling and turn on cell division and so on. But it's well controlled. When the KRAS has a mutation, and all of a sudden the cell will be constantly being turned on. So you want to actually find a drug to target this mutated KRAS. But it turned out that, that even if you have a drug to target the mutated KRAS, in cancer cells, there will be continued mutation. So whatever the drug that you initially developed may not work. So many people are thinking, well, maybe this is not a good target. Let's find other target. So along the pathways of the MAPK pathway, you can all of a sudden find all of these proteins along the ways, and then there are many, many drugs. And many people are arguing, well, my drug is better, and then my drug is probably weak with a certain side effects. But I think that this is what I meant, generate disease biology insight. In order to generate something like this, you need, of course, a biology expert. But now you can also learn it from omics data, maybe doing some single-cell RNA-seq analysis. Or maybe you can actually create a database and then try to learn it from the large database. Or you can actually mine PubMed articles. And most recently, I would argue that if you have especially custom-trained AI agents, and that could actually handle a lot of the information, except for maybe it doesn't do the data analysis for you, right? And so KRAS is highly mutated. If you actually just look at the COSMIC database, and you see that the highest bar is the mutation rate in pancreatic cancer. That's 60% of the pancreatic cancer cases have this mutation. So obviously you would say, well, this is a wonderful drug target, and what's wrong with it? Well, what's wrong is actually it's the engineering issue. So once you identify the drug target, the story of drug development just got started. There are many, many drug modalities that you can consider. For example, depending on different mutations, and then there may be slightly different chemicals that you can use. And sometimes you don't want to directly address the mutation site. Instead, you want to actually block the KRAS and other protein interactions, and that could be more effective and mutation resistant tumors, and you may also be able to get away with that strategy. Or you can actually find the pharmacocore where it's located in the GDP and GDP reaction site to compete out of the substrate. So there are many, many modalities that you can consider, and each of the modalities require lots of knowledge on actually the engineering, bioengineering or the chemistry. But one point that I would like to mention is that sometimes you do have to get beyond the target gene concept. And there's an article published just 2 years ago that talk about another immunotherapy drug target. And just by focusing on the target, and if you were trying to improve the precision of maybe predicting the immunotherapy response, it's not very good. However, if you consider all of the neighboring, the so-called protein interaction network modules, and try to build your predictor, then all of a sudden you can actually have really much, much better performance. So that really means that you have to take a system approach as opposed to just focusing on the disease biology or the engineering in order to win this game. And this is why I think I want to emphasize that if you want to actually find potential new treatment, you have to actually find novel target. And finding novel targets sometimes is you have to take a database such as KRAS and MAP signaling, MAP kinase signaling pathways. Here's I'm showing an example, a screenshot from the pager database where I showed a pathway that's called a RAS signaling, and it has a size of 316. And you can see that the KRAS is only ranked 25 according to network topology. There are many, many other potential targets that you could consider, but maybe they're not variable because they're not highly expressed in tumor, they're very bad, but then at least you should consider them. That's the point. Now, what is the AI's role in this whole process? I think AI is really being popular as efficiency boosting software tool. Right? When you look at the entire process of going from target to initial heat compound identification, lead optimization, clinical trials, preclinical trials, clinical trials, and real-world post-market surveillance. And AI has been injected into every step of the process. For example, there's one paper that talks about having initially a space of all the candidate molecules. If you use AI and do transfer learning and try to take into account of the information, the context where this molecule is being targeted, and you can actually shrink your space and find maybe better lead compound or even optimize the compound that can actually target. So here you would, there's another example from a company that is actually raised billions of dollars called in silico medicine based in China. And they've been able to take an existing drug target and come up with their proprietary platform called the Gento. And that Gento can come up with the chemical compound de novo and then figure out the synthesis pathways. And then this whole process, according to the Nature Biotech articles, took only 46 days as opposed to traditionally would be a year or 2 years. And that was pretty impressive, right? So I think recently I heard that for this exactly the same compound, they were able to enter a $1 billion multi-phase contract with a major pharmaceutical company. So that's good for them. Now, what is the realistic goal for the Spark Center, right? I think the realistic goals is if we're talking about the data set, how about just curating the data set that has multimodality, multi-skill data? And we want to make it open. We want to make it so easy to use by potential drug development researchers. And when we are talking about model, and there are different modalities of drug discovery, and each modality, maybe we can create a model. And these models are what I call the digital twins model. And then essentially, we are not just setting up a parametric model that traditional engineer used to do, but digital twins model differ from the traditional engineering model in the sense is, first of all, it's multi-tiered. It's closer to the real world. And the second is it's data rich, and it's different from the parametric model that doesn't have data populated into it. And then I want to talk about the center want to use strategies, essentially thinking about the drug development and therapy as a programmable drug development process, and I'll elaborate a little further. And then we want to nurture an ecosystem where everyone thinking about drug discovery, maybe have certain missing skills, can come together to this center and then learn from each other. And we will have exchanges and we'll share the different tools together. And then I would also imagine that this center will also become a place for education and training. Informaticians can actually use this as a place to talk to clinicians, chemists, and basic science biological researchers. So we roughly followed from data to network pharmacology models to digital twin simulations, and then in the end, personalized therapeutics. I'll show you some of the work that we're doing. So we currently are developing Pharma Alchemy 1.0 database, and it's already been presented last November in Genome Informatics workshop in Singapore. So in this database, we not only have comprehensive knowledge curated for gene, drug, and diseases, but also about their connections and relationship. And if you're interested, and you can talk to Kevin and Melissa sitting up front. And actually, Kevin is going to do a talk later today about the Pharma Alchemy database. And not only that, we're also trying to populate the data with very deep information that has not been generated before. For example, we are funded by this project called the Cell Models for AI. Essentially, we're working with researchers from Stanford, UCSD, UCSF to generate deep perturbation level data for iPSC cells, and that may have neuroscience and cardiovascular disease implications, and also a triple-negative cancer cell line. We're capturing both the protein-protein interaction information and also the spatial proteomics information using imaging, imaging, microscopy. We're also doing the genetic perturbations, and we put all of this information in a structure that is easier for machine learning and AI. In fact, there will be a so-called a jamboree in March if you're interested in learning about this data set, and talk to me. Okay? And then we also think about collaborating with internal researchers. Rob Wellner, my collaborator, is very interested in NK cells. And so he did experiment the NK cells harvested from bone, spleen, and blood. And we were able to not only do the traditional single cell analysis, but we were also able to chart the network level changes when the cell developed from one state to another. For example, if I just move my mouse over, you can see the up and down regulations of genes in this network. So it's this kind of a representation that is really good for us to extract the disease biology and make it easier to potentially find the drug target. Right? And then another example is work done by my postdoc, Asam. So we were looking at several drugs, and then we were looking at the representation called the gene terrain, and longitudinally, right? So drug W seemed to have some effect, and then, but the drug M doesn't seem to have any effect. But when you put the two drugs that doesn't seem to have obvious effect together, T and M together, all of a sudden you see a synergy effect. So it's this kind of a technology that actually can show you a lot more profound information than a simple phenotypic readout. And we're really excited about this information because this essentially gives us the model that can also connect it to potential simulations. If we can learn from the experiment all of these the changes and the conditions, and then we can potentially build a model that are predictive. And I also want to actually come back to the point of programmable therapeutics development. Think about the New York subway, right? So the gate is closed, and no matter how hard you push it, it's going to remain closed unless you pay or show the ticket. So they actually have a locked and unlocked state. Depending on which state the gate is in, even if you try the same method, which is to push the door, it's not going to work in one case and it's going to work in another case. So likewise, right, this is my thinking. And for cancer, there's going to be sensitive and resistance. I'm just very broadly just use this for illustration. Of course, there are more states than that. In the sensitive state, the cancer is going to actually work. The drug A is going to work and then killing the cancer. But at some point, the drug A is actually killing enough of the cancer, but then a very small clone is going to be resistant, and it's going to become this state, resistant state, where the drug A no longer work. So it's like all of a sudden the cancer locked up. And then now the key here is you have to find another drug to somehow return this resistant cancer into a sensitive state. This is the key to what I said, the programmable medicine. But the key to this is also being able to not only see the simple state at the phenotypic level, but also at the molecular level, which is what we're using gene terrain for. Right, so in programmable medicine, I think is going to be the key to achieving the peace mission of the center. Because you can think is essentially if you can read out the diseases in real time, and that will be very expensive. Many functional genomic assays, many pathology or imaging assays, but let's say we can do it. Right? If we can do it, now it's like gaming against the cancer in the chessboard. Now, if we can find out all the optimal paths or even predict the cancer's next move, then the medicine development is no longer as difficult as a $1 billion 8 to 10 years mission. Now it should be as easy as using what we have and only inventing the new method as needed. A lot of the time, maybe we don't need to work as hard. Right? So I think I still have time. Thank you, Jim, for buying me some time. So I'll show you this example, right? So in this particular example, we're using my collaborator, Dr. Chris Willey, and we had U1 funded in the past several years to develop glioblastoma patient-derived xenograft models. So we had a lot of fun with this data, even though the original objective of the grant was just to develop the model and not to explore the application. So one of the model is X1516-1, right? So this model, we put it in an embedding space, which is essentially a low-dimensional projection of multi-dimensional data. So it's a fancy way of doing clustering. So we clustered this PDX cell line in the context of all the TCGA glioblastoma tumor. And then there are some other PDX that's spread out. When we look at this neighborhood, we found that the neighborhood roughly is in a very low survival neighborhood. So essentially, if you think about this as a chessboard, this is a chessboard that have marked as poor survival, really bad, run away from this point, right? So we look at the radiation. So there's a post-radiation, and then it doesn't even move, right? So that really means that this cell line is the radiation-resistant cell line. It does not move, and that's why people are having such a short survival given the standard of care, right? It's something that we need to do something to make it radiation sensitive. If you think about the programmable medicine idea, and then can you come up with a drug to actually make it radiation sensitive, as opposed to just coming up with a drug that kill it off, and then maybe several $8 billion later, and then you may find it. So here's our solution. So we'll look at the heterogeneous data from, I think, 11 GBM patients, single cell level resolution. And we're looking at all of these actual patients and their single cell, right? So single cell, they're all, I think, the brain, I don't remember what exactly the brain cells. But then we're looking at the single cell level resolution. We're looking at gene terrain as a representation of the cell signatures. You can see some of the cell signatures clustered together. They really share the similar gene terrain images. Whereas if you move away, and then they have very different signatures. In fact, there's another sensitive cell line that move its spot from a low survival spot to a high survival spot. So there are so many data points that we can actually learn by showing what are the experimental paths that will trick the cell to go from one spot to another spot. And once we can learn enough of these examples and we can build predictive models, and the hope is that we will be able to program the future of cancer drug development using combination of these gene terrains and omics data and drugs. So there's still a lot of opportunity for drug AI breakthrough. These does not involve coming up with new ChatGPT. So for example, you can think about the ChatGPT when it's trying to solve a bioinformatics problem, it probably just use a black box AI. It doesn't try to tell you the mechanism because the mechanism hasn't been discovered. Unless ChatGPT can read the single cell analysis data, then I think all our department need to dissolve by then. I don't think it happen anytime soon. So a lot of our work, and we're interested in having faculty recruited into the center and department to do explainable AI, especially explain what's happening at the molecular level. And reasoning from rules only, this is what the ChatGPT is doing really well. But can you reason not only from the rules, but also from the data? That is a different type and instance jump. And instead of doing connectionist approach, just like ChatGPT, can you do neurosymbolic AI? Many of us probably have the experience of using ChatGPT, try to generate some figures, and the figure is fine, but all the words on the figures always have misspelling, right? Have you noticed that? That is the reason because ChatGPT cannot do symbolic AI and the connectionist AI at the same time yet. Parameter-rich versus data-rich, I've showed you that gene terrain is data-rich models. If you do parameter-rich models, that's the traditional engineering model. One-level model versus multi-level model, our human system is so complex from physiological level down to the subcellular level, we really need to integrate all of them. And then I'll skip that and I'll say, well, we really want you to participate, right? So you can apply to join a member. You can actually recommend amazing staff and faculty to us. You can collaborate and, in a drug discovery project as we're building all the different components, or you can actually help us make up our weakness and shortfalls on understanding clinical trials, because we do need to convert from in silico trials to clinical trials. The last point is we're having a more in-depth discussion on this topic in this year's ADIS to be held on May 3rd. And the theme is when drug discovery meets AI. Does anyone know what this poster is copied after? There's a movie. What? What movie? When Harry Meets Sally. Yeah, that was one of my favorite. But anyway, so that's what we're trying to do. On one side, and the medicine, on the other side, the people from the department. And I really thank all these people listed here in my lab. And I'll take a few questions. No questions? Okay.
Okay, thank you, Jake. It's inspirational and a little scary when you said we could all be replaced by AI. Hopefully we've got a few years left, but we'll see. Last I want to end with a presentation on our educational program. As I mentioned, we have a Bachelor of Science program in Bioinformatics that we were actually charged with creating that program when the Institute started in 2015, and that the development and management of that has been ably led by Elliot Lefkowitz. But now we've been trying to work within existing PhD programs and really found that the interdisciplinary nature of biomedical informatics and data science doesn't fit well with the existing programs. There's too many other things people need than just things offered by sort of the GBS or biomedical engineering. And so we're now preparing our own PhD program. And Jin Chen, who came to us last year from the University of Kentucky, is actually in the Department of Medicine in nephrology. And he's an informatician, and he came to me and said, is there anything I can do in informatics? And I said, oh yeah, what do you think about educational programs? And he was all over that. So Jin, you want to come on up and tell us about the developing PhD program?
So welcome everyone. Today we're embarking on an exciting journey as we're introducing the new Department of Biomedical Informatics and Data Science. In the next couple of minutes, I'm going to introduce you the vision, the plan, and the current status of our educational program in this new department. So biomedical informatics is by nature an interdisciplinary field that combines many domains like biology, medicine, computer science, statistics, and so on. It's about harnessing the power of biomedical data to revolutionize health and research. As Jim just said, biomedical informatics is not just about applying any existing algorithm to bioinformatics data. It involves many aspects like information theory, knowledge, algorithm developer, and many other things. And the sole purpose is to advance the informatics research. So to inspire our new generation to join us and shape the future of biomedical informatics, we'll develop a series of educational programs in biomedical informatics, from certificates to PhD programs. We'll commit to prepare individuals for the development and application of informatics theories, tools to solve problems across life science and health ecosystems. So our educational efforts emphasize analyzing and interpreting complex data and developing and disseminating solutions, infrastructures, and algorithms to address human health and disease problems. By doing this, we will grow skills and professionals who are not only proficient in cutting-edge computation technologies, but also deeper knowledge in biomedical and health sciences. That we trained to lead interdisciplinary research, innovative with data, and science-driven solutions, and build a real-world infrastructure. So our graduates will also navigate the ethical, legal, and social implications of informatics and AI theories methodologies to ensure the responsible and the beneficial use of AI. So through this comprehensive educational approach, with also the programs ranging from certificates to PhDs, were committed to advance healthcare, improving patient outcomes, and ushering into a new era of medicine in AI. So AI in medicine or in health informatics is about the real data. The real data biomedical application and the practice forms the cornerstone of our educational approach. And where the theory knowledge of AI and informatics is actively applied to meet the tangible challenges, health systems, and the biomedical research that we face today. In these diverse professional settings that form the clinical laboratories to research institutes, the ability to translate complex algorithms, data-driven insights into actionable solutions, the students are expected to learn how to apply knowledge, AI algorithms, theories, and information solutions to address tangible challenges in real-world healthcare and the biological research problems. The biomedical informatics is inherently interdisciplinary. It includes knowledge, methods, tools from stats, biology, medicine, AI, and health informatics. To that end, we're going to develop a curriculum that span multiple departments at the UAB. The students will master data analysis pipelines, including data harmonization, data mining, data modeling, and interpretation. So all these are critical aspects of data science. They will form the backbone for our research advance, providing the key to unlock the answers to complex biomedical and healthcare questions. So by traversing the boundary of traditional data disciplines, bioinformatics informatics students will master diverse tools and methodologies from this field to catalyze innovation, inform clinical decision-making, and enhance patient outcomes through data-driven evidence. The educational program emphasizes the importance of team science and also the consideration of the social implications, which form the environment where the collaborative research projects span multiple disciplinaries. By encouraging students to work in multidisciplinary teams, the program aims to harness the collaborative expertise necessary to tackle the complex ecosystem. So our current efforts are focused on the development of the Biomedical Informatics PhD program in collaboration with departments of medicine and the Health Services Administration. Our PhD program is designed as a core track model, which means the students will first take the same sets of core courses. These courses will provide the students a solid and a broad foundation of a PhD level analysis, knowledge, and skills. And then the students will select a track for which they want to deepen their understanding and their knowledge in informatics. Those tracks include translational bioinformatics, clinical and health informatics, and AI medicine. To ensure the transdisciplinary of the degree, students choose one of the courses as their academic path. But additionally, students are encouraged to take a variety of elective courses that's offered by the other programs at the UAB. Students will learn the knowledge and the skills from these elective courses to support their PhD research. This will also extend their knowledge and skills acquired from the core and the track courses. So you can see the future of biomedical informatics is in the hands of our students. And the way we educate and nurture them today will significantly shape the trajectory of this field. By providing them with a robust educational foundation, instilling in their commitment to interdisciplinary collaboration, and establishing the understanding of the ethical, social, and clinical implications of their work, will effectively equip the next generation of innovative leaders. So just to remember, our role in this department is not just guiding these brighter minds, in the academic, not just about the academic responsibility, but the crucial investment in the future of the healthcare and the scientific discovery. Thank you.
So, you know, one of the things that we're still developing is the actual specifics of this interdisciplinary program. And so, you know, in a lot of places that have informatics programs, there's a bioinformatics program, there's a clinical informatics program, and there's often very little overlap. But given, you know, the world today where we've got personalized medicine, people who are practicing medicine need to understand genetics, people who are developing clinical informatics applications and theories and tools need to understand bioinformatics theories and tools, and I think vice versa. The people that work in the bioinformatics end of the spectrum need to understand that we're not here to try to improve the lives of zebrafish and fruit flies. We're here to improve human health and disease. And so understanding what the findings in the laboratory are, what their implications are for patient care is going to be important, and understanding, of course, how to bring genomic medicine to bear to clinical medicine is important as well. So all of our trainees are going to have a common core, and everybody's going to learn some bioinformatics, and everybody's going to learn about electronic health records, and everybody's going to learn about something about the things in between, so that as they move forward, they will be able to understand how they're going to contribute to this spectrum of biomedical health and informatics. Okay, well, that concludes our formal program. We're going to have a closed program now for folks that are just in the department, so we can get to know each other better. We've had a number of additions since the beginning of the pandemic, and a lot of people have never met each other, including me. There are several people I haven't met in person because they've been working remotely or hybrid. So we're going to get a chance internally to hear about what each other's doing. The rest of you, thank you for attending. I will put my slides up. We'll get Jake's slides and Jin's slides, and we'll get those up on our website. I think there's a video. I shudder to think about looking at that video, but I think we'll have that up there as well. If you missed something and want to go back to it. Check out our website, still in development. We are still in the process of getting our jobs posted, but once they're there, let your friends elsewhere know. If you have folks that you want them to move to Birmingham, tell them about the opportunities in biomedical informatics and data science. With that, I'll conclude the public part of our program, and thank you for your attendance.